All right, so we're looking at chapter 3, part 3, and we're going to dig in a little bit further into amino acids, peptides, and proteins. Specifically, we're going to be looking at methods to characterize peptides and proteins. So if we think about how we name peptides, you're always going to start at the N-terminus. So if we were to use their full amino acid names, that gets kind of clunky and becomes less useful. So typically we're going to refer to peptides by their 3-letter code abbreviation, or if it's a longer peptide, sometimes we'll use our 1-letter code. And if we think about all the different places where we find peptides, we're going to see them as hormones or pheromones. So if you think about insulin or oxytocin or sex peptide. If you think about neuropeptides, so substance P as a pain mediator, or antibiotics, or for protection, say for example toxins like amantin, or conotoxin, or chlorotoxin. These are all examples of where we find proteins. And it's useful for us to recognize that polypeptides, which are these covalently linked amino acids, also may involve cofactors, so something that's a non-amino acid component, could involve metal ions or organic molecules, as well as incorporating coenzymes, so organic factors, or something like NAD+ in the lactate dehydrogenase. And then if we think about our prosthetic groups, so these are covalently attached cofactors, say for example, like the heme in myoglobin. And then there are other types of modifications. So if we think about that, that's a pretty large collection of different types of molecules. And so it might be really valuable for us to think about how we might go about separating these to be able to isolate the compounds that we care about. So if we think about a mixture of proteins that could be separated, there are several different types of functionality that we could use. So whether that's a physical or a chemical property, so we might look at charge or size or an affinity for a ligand, or perhaps solubility, or maybe hydrophobicity, or their thermal stability. And so if we're doing just a preparative separation, chromatography is a really great place for us to start. So if we take a look at column chromatography, essentially we have a reservoir. And over time, what we're going to see is as we draw a solvent from our reservoir, and we've loaded our protein sample in the mobile phase, and then we have this solid porous material, what we're going to see as we pump that solvent through is that we're going to start to see separation. And largely that separation is going to be due to polarity. And over time, eventually we will actually have those compounds come off the column, and we'll be able to collect them separately in a fraction collector. And then using a detector, we might be able to identify what type of molecule or molecules we've collected. Now, one of the really interesting things and what's important to think about here is if you don't set up your column appropriately, you can actually have everything move through all at once. So part of what you're looking for are idealized column conditions that give you the greatest possible separation between the types of materials that you're trying to identify. If we think about separation by charge, this again uses a column, but now instead of separating by polarity, now we're actually looking at charge. So we have our protein mixture that's added on top of the column, but then within the column itself are what we call cation exchangers. So you have a large net positive charge, you have a net positive charge, a net negative charge, and a large net negative charge. And so these proteins then move through the column at rates determined by their net charge. So whatever pH is being used. So if you have cation exchangers, proteins that have a more negative net charge are going to move through faster and elute earlier. And so we use these polymer resins to control the type of charge and charge interaction, which allows us to be able to separate out proteins that have different charge species. If we're separating by size, it's a very similar principle, except now we have these cross-linked polymers that allow our molecules to move through. So larger molecules pass more freely, appearing in the earlier fractions, whereas the smaller molecules move more slowly, and it is these porous polymer beads that determine that size exclusion separation. And then there's affinity. So this is a slightly different column where if you have your protein mixture that's added to a polymer, and on top you're adding your protein mixture, but within that polymer, there are ligands that have specific binding sites for certain types of proteins. So you're going to pump your ligand, right, through your column, and then what you're going to see is that there are specific proteins that bind to that specific ligand. And so that's what we refer to as affinity chromatography, because it's the affinity of the ligand for the protein that allows you to separate out what you're looking for. One of the most common ways that we're going to see protein analysis occur is through electrophoresis. So this is separation usually for analysis, and it's where we use an electric field to separate proteins according to their charge. So we have a gel matrix that stops proteins from moving based on size and shape, and we apply a current, which then allows only, right, our samples to be able to move based on the direction of the current. So if we think about this, here's an example of gel electrophoresis. This is the direction of migration, right? We actually load our samples, and then we can actually use a detergent, SDS, which is sodium dodecyl sulfate, and that's going to cause our proteins to unfold, and that gives them a uniform negative charge, so the native shape of the protein doesn't matter. And so that means then that the rate of movement is only going to depend on size, so our smaller proteins are going to move faster. And so if we take a look here, right, these are our markers, right? We have our uninduced cells, we have our induced cells, we have our crude extract, we have a precipitate, our anion exchange, our cation exchange, and then our purified protein. And that's what's going to allow us to be able to analyze, say, for example, a very complex mixture. Now, we can use our SDS page to calculate the molecular weight of a protein. So if we have an unknown protein and we have our relative standards, we'll be able to determine its relative migration. We could also use isoelectric focusing to determine the isoelectric point of a protein. So that's where I apply a protein sample to one end of a gel strip with an immobilized pH gradient. And then I apply an electric field. And after staining, I'm able to determine the distribution along that pH gradient according to the isoelectric values. What's brilliant about this is that you can actually combine isoelectric focusing and SDS page in two-dimensional electrophoresis. So I take my protein sample and I first separate them on a gel strip using isoelectric focusing, and then those separate proteins are separated further in a second dimension on an SDS polyacrylamide gel. And so what you can see here is that you go from left to right, there's decreasing isoelectric points. And as you go from top down, right, you have your decreasing relative ratios compared to your standards. In addition to the 2D electrophoresis, we also have the ability to use spectroscopy in order to determine our aromatic amino acids. We talked about that briefly when we talked about the Beer-Lambert law. So because aromatic amino acids absorb light in the UV region, they have this absorbance maxima somewhere around 280 nanometers. Tryptophan and tyrosine are the strongest chromophores, and so we can use concentration as well as the spectrophotometry with Beer's law to be able to determine whether or not we have aromatic compounds. And as you can see here, this is yet another example where I have wavelength, I have my absorbance, and you can actually see the difference in terms of the absorbance of tryptophan as opposed to tyrosine compared to phenylalanine. We could also use specific activity. So this is the activity per total protein in order to determine protein purity. So to figure out how much of the protein that's active that we have. And then we also might be interested in protein sequencing. So once we've isolated a protein, it's important for us to be able to determine the sequence of amino acids of that protein. Now the sequence itself is usually determined from a DNA sequence. So we can use the Edmond degradation, which is a classical method. So this is where I do successive rounds of N-terminus modification. So I cleave, right, identify that single amino acid, and then I cleave again. And so this is what can be used to identify a protein if there's a known sequence. I can also, though, use mass spectrometry, so MALDI-MS and ESI-MS. I can use those to be able to identify the mass of a peptide and the amino acid sequence. And so if I have post-translational modifications, this mass spec method might be a more accessible. So if you take a look at Edmond degradation, here's kind of that brief summary of what's happening. I have my polypeptide. I have a high pH in my phenylisothiocyanate. That's going to form a carbamyl derivative. And then I undergo, right, this cleavage at low pH. And now I've shortened my peptide by 1 amino acid residue. And so as long as I can identify the amino terminal residue of the polypeptide, I then purify and recycle the remaining peptide fragment through the Edmond process again until I've isolated each of the amino acids. If we want to think about what the mass spectrometry procedures for the sequence IDs are, so here I have a sample that I inject, and what I'm going to see is that, right, in my mass spectrometer, my masses are going to separate based on size, and then I'm going to see detection. And what I'll observe is this fragmentation pattern. And so I'll be able to determine, right, the components based on their relative mass. And so you can see here's relative signal intensity, here's the mass. And so based on a close-up view of this, I would be able to determine, say, for example, serine or alanine based on its mass. So these are all methods that we use to be able to determine not just the identity of the protein with respect to its amino acids, but also to be able to separate mixtures of proteins, which might be valuable, say, for example, if you're doing a cell assay and you're trying to determine what the protein of interest is. So these are very practical ways that we would be able to do analysis of proteins, protein mixtures, or amino acids. Thanks.